everyone, and welcome to my channel Study with YT, where I will be posting educational content for high schoolers who want to supplement their studies with some further free online resources. This video is a follow-up to my previous video on Section A of the VCE Biology 2019 exam. Here I will be going through Section B, which is the short answer section worth 80 marks in total. A couple of disclaimers before we get started. Firstly, the following exam paper was the test conducted on the 1st of November 2019. All contents are copyrighted to the Victorian Curriculum and Assessment Authority 2019, and it is freely accessible by anyone with an internet connection from the link listed here, and I'll make sure to also leave the link in the description below. Question 1, worth 4 marks in total, starts with two diagrams of molecules that are required for the production of proteins within a cell. Molecule 1 is a single nucleotide chain, so this must be RNA. Specifically, this is a transfer RNA given its shape. Molecule 2 is a double helix structure, so this must be DNA. Part A of the question, worth 2 marks, asks you to name two differences between the monomers of the two molecules. Now, to quickly compare RNA with DNA, RNA is a single nucleotide chain and it is composed of a ribose sugar and 4 nitrogenous bases, which are adenine, guanine, uracil, and cytosine. DNA, on the other hand, is an anti-parallel double-stranded helix, and the DNA nucleotides are composed of a deoxyribose sugar, phosphate, and its nitrogenous bases are similar, except instead of uracil, it has thymine instead. Now, going back to the question, it is very important to answer the question itself rather than info dump onto the exam paper. The question is not asking you for the difference between DNA and RNA, but the differences between the monomers of the molecules. So the two differences you should give are ribose sugar versus deoxyribose sugar and uracil versus thymine. If you couldn't remember the nitrogenous bases, you could also specify that RNA has a ribose sugar, so it in theory has more oxygen molecules compared to the deoxyribose sugar in DNA, and this was also an acceptable answer. The other comment that VCAA made was that the spelling was very important. Some students had put thiamine, the vitamin, down instead of thymine, the nitrogenous base. Only 18% of the cohort got full marks, with over 50% of the students not getting any marks. Genetics is a huge core topic, and this is a knowledge recall question, so you should be able to nail this question down. Part B of this question, also worth 2 marks, gives you 10 amino acids shown here. It then goes on to state that the translation of these 10 amino acids was found to contain over 300 monomers. The question asks you to explain why there can be over 300 monomers for only 10 amino acids. This question is testing your knowledge recall regarding gene expression. A quick refresher, gene expression involves two major stages: transcription in nucleus, then translation in the cytoplasm. In transcription, messenger RNA is synthesized from a DNA template. The RNA polymerase enzyme makes the mRNA according to the DNA RNA base pairing rules. A typical gene will consist of a promoter segment, a coding segment called exons, and a non-coding segment called introns. This then goes through a post-transcription modification where mRNA is modified by enzymes that cut out the introns and piece the exons back into an mRNA. This then leaves the nucleus into the cytoplasm. In translation, mRNA is read and a polypeptide product is assembled. Here, codons, a 3-base code, helps tRNA identify the correct amino acid to bring to join together to form the polypeptide, which will then fold to form a complex 3D structure determined by its amino acid sequence. This completed polypeptide is then released into the cytoplasm. For 2 marks, you have to provide 2 facts, which are, firstly, each amino acid is coded for by 3 monomers. So this counts for about 30. This counts for exactly 30 monomers of the 300. The remaining monomers are made up of introns, which are the non-coding segments of mRNA. The clue here that the paper provides is that this section of the RNA molecule was found in the nucleus of the cell, which means that the introns are still within the mRNA. The breakdown in terms of marks for this question was pretty similar to the previous. Question 2 shows you a diagram of a chloroplast, which the question states is surrounded by a double membrane. Part A1 asks you to name two molecules that are inputs for photosynthesis. This is a straight knowledge recall question. And the answer is water and carbon dioxide, and most students did well. Part A2 states a higher concentration of oxygen is found in region R as annotated on the diagram when a plant is photosynthesizing compared to when it is not photosynthesizing. It asks you to account for the difference in oxygen concentration found in this region. Here, for 2 marks, you must identify correctly how oxygen is being produced during the light-dependent reaction, the first stage of photosynthesis, which is water is split to produce oxygen gas. For the second mark, you need to state why oxygen would be found in region R, which is the stroma, because remember the light-dependent reaction occurs in the disc-like structures called the grana. So you need to also mention that the oxygen produced then diffuses into the stroma. VCAA was also quite generous that they would give you a point if you mention when light is not available, oxygen is not produced. Part B of question 2 worth 2 marks asks you to describe the role played by each of the coenzymes NADPH and ATP in photosynthesis. This is another knowledge recall question. Remember, during photosynthesis, water in the presence of light and in chlorophyll is converted into hydrogen ions, oxygen gas, and ATP. The ATP and the hydrogen ions are then combined with carbon dioxide to be converted into a glucose, water, and ADP. The NADPH coenzyme is what helps transfer the hydrogen ions, and the ATP transfers the energy for the light-independent reaction, which converts the carbon dioxide into glucose, water, and with the energy expended, ADP. Question 3 starts with some information regarding the human immune system, so go ahead and pause this video to have a read and recommence when you're ready. Okay, so part A asks you to list 2 possible routes the bacteria could use to access the living tissues of the body. This is knowledge recall, 1 mark for each correct route, and I've just listed some here as examples. Overall, this was a fairly well answered question. Part B states that there are some chemical barriers to these bacteria before the cells are even aware of the bacteria presence. It asks you to list one of the chemical barriers and explain its function. So for 1 mark, you need to correctly identify a chemical barrier and then explain how it acts as a defense against bacteria. Once again, I've listed a couple. You can find other examples as well. Generally, it seems students either got this question or they didn't, with only a small proportion of students getting only 1 of the 2 marks. Part C asks you, how does histamine contribute to the inflammatory response? Once again, straight knowledge recall. You can list any of a whole heap of responses of what histamine does. I've just listed a couple of examples here. Part D asks you to describe how an adaptive immune response is initiated during a bacterial infection. This is another straight recall, but make sure you answer the question directly. The key word here is initiate. So talking about down the track things such as cytotoxic T cells will be incorrect. You need to talk about the initiation process. In this question, that will be about the antigen presenting cells, or APC, displaying antigens on the cell surface. This then interacts with the helper T cells, and subsequent steps in adaptive immunity does involving cytotoxic T cells, but talking about them in this question will not score you points. Question 4 shows you a diagram regarding how thunderstorm asthma occurs. So pause this video to have a read and recommence when you're done. Okay, so part A asks you to name the cell and protein that is involved in allergic reactions. This is knowledge recall, and the answers are mast cells and IgE antibodies. Part B asks you to list 2 strategies that a person could take to reduce their risk of developing thunderstorm asthma. Obvious strategies including reducing the exposure to the pollen fragments. So you can write about staying indoors to reduce exposure, wearing face masks. You could also talk about reducing the body's reaction to the pollen fragments by taking antihistamines. Part C asks you to explain if it could be possible to develop a vaccine against thunderstorm asthma. Now, this is a question where you could argue either way, as long as you can justify your answer using knowledge regarding how vaccines work and how the body develops immunity from the vaccine. So students could argue that there are too many different pollen fragments for scientists to develop a vaccine with, as there is no single identifiable universal component that they could put in a vaccine for humans to mount an immunity with. And if they put all the pollen fragments into a single vaccine, then the response that you would have to mount in order to gain immunity would be really high. However, if you want to argue that yes, it is possible, you may have been taught in class or have personal experience with something called desensitization injections for allergies. But one way you could switch off the body's immune reaction to pollen fragments is by slowly, over time, administering incrementally higher doses of the allergen to a patient. Once their body is able to cope with the allergen at a level much higher than you would expect to experience in real life, then their body would not mount a response during a thunderstorm asthma. Either of these answers would be acceptable by the examiners. Question 5 starts with some information regarding the beak depth and length of Galapagos finches, so pause this video while you read it and recommence when you're ready to go through the questions. Okay, so part A asks if BMP4 and CAM are both highly expressed, what beak shape would you expect? Now, if BMP4 is high, you're looking at that second row, and given that BMP4 affects the beak depth, you would expect it to be deep. If CAM is high, we're looking at the third row, and since CAM affects the beak length, you would expect it to be long. So the answer here would be deep and long. Part B introduces the gremlin gene, which inhibits the expression of BMP4. The question asks if this gene also affects the cells of the beak, what would be the end result? Now, this is a 2-mark question, which means you need to give 2 statements. The important thing here, which I think some students missed out on, is that the question even gives you a clue. What would happen to the beak shape, which is depth and length? So the answer here is that the gremlin gene inhibits the expression of BMP4, so the beak depth would be shallow. That's 1 mark. The second mark is that the gremlin gene does not affect the CAM gene expression, so the beak length would be unaffected. You need to state both of these sentences in order to get the full marks. Part C asks you to explain why a mutation that affects the expression of BMP4 gene can lead to rapid changes in the phenotypes of Galapagos finches. Now, this is a 3-mark question, so you need to make 3 statements. Most students can identify that this is a natural selection question, but in order to answer the question properly, always make sure you answer the question asked directly rather than blurt out a prepared natural selection answer. As you have learned from your studies, BMP4 is a master gene, which means its expression affects many genes. Statement 1. Any changes in BMP4 expression will subsequently alter one of those genes, which may give the finch a new selective advantage phenotype. And as they survive, produce, and pass the new trait on to a future generation of finches, this could lead to rapid changes in the phenotypes of these Galapagos finches. Question 6, part A1 asks, what does aneuploidy refer to? This is a knowledge recall question, and the answer is an abnormal number of chromosomes. The fetus may either have more or less chromosomes than normal. Part A2 asks what would scientists examine in the woman's blood sample? The question stem is where the answer lies. It tells you that this test is used to detect fetal aneuploidies. So you have to specify that. You have to specify that A, it is testing for fetal rather than maternal, and that B, aneuploidy means that it is testing for fetal DNA or chromosomes. Answer of anything else such as maternal blood, maternal DNA, or maternal chromosomes would be incorrect. Part B asks you to describe 2 different types of block mutations. I've listed a few, and any 2 of these will do. The important thing here is to limit your answer to genetic abnormalities, ACA related, as in these have to be related to the chromosomes. If you start talking about nucleotides or amino acid changes and not related to chromosomal mutations, then examiners will not give you points for those answers. Part C asks you to translate the sequence above using the codon table listed here. The answer is as shown. Part D1 asks that if the 20th nucleotide changes to uracil, how will this alter the amino acid coded for in this sequence? So referring back to the table, you'll see that by changing the 20th nucleotide from adenine to uracil, the 7th amino acid would change from GLUT to val. Part D2 asks that if the 20th nucleotide was deleted, how would this mutation alter the structure and function of the hemoglobin protein produced? This, as you know, will result in a frame shift. So all amino acids from this point onwards would be different. Relating this back to the question stem regarding sickle cell anemia, this means that the resulting hemoglobin protein would be non-functional and its structure would be different. Question 7 shows you 3 different species as well as their size. Part A asks you to identify what type of evolution is involved for these 3 species to share a common ancestor, yet have completely different features. For 2 marks, you need to identify correctly the type of evolution involved, which in this case is divergent evolution, and that by being exposed to different pressures from their environments as well as other species around them, both competitors, prey, and predators, they have evolved to end up with completely different features. Part B asks for one possible cause of a species extinction. You can select any of the following as your answers. I put ice aging here as well because the question stem mentioned the specimen was frozen. Part C asks you to describe what would have occurred to lead to the preservation of an animal as a specimen. I've listed the following steps. The step bison would have had to die and be frozen prior to any predators or scavengers being able to eat it. By being frozen and not exposed to the outside environment, the corpse would not be subject to decay, and over time, once the ice has melted or eroded, the specimen would have come closer to the surface and be exposed for discovery. Question 8 has a long question stem, so please pause this video to read it and press play again when you are ready. Now, Part A asks you to give an example of a disorder or disease that can be detected by genetic screening. I've listed a few here, and you may have other examples that you've learned in class that you want to use instead. Part B asks you to describe the 3 stages of PCR in the correct order. Now, a bit of a refresher. So polymerase chain reaction, or PCR for short, is a technique that amplifies a piece of DNA using a chain reaction of DNA replication events. As a result, a large amount of DNA can be produced in a test tube in a few hours. There are 4 main ingredients that are required: a sample of DNA, a source of 4 nucleotides, the Taq polymerase, which is the heat-resistant enzyme that is used in PCR, as well as single-stranded DNA primers, which help specify the starting points of DNA replication. The ingredients are then placed together and heated up to 90 to 95 degrees Celsius. This helps break down the hydrogen bonds and separate the strands into 2 single-stranded templates. The thermal cycler then cools to 50 to 60 degrees Celsius to allow the primers to bind to the complementary DNA sequence. The temperature then increases to approximately 72 degrees, which is the optimal temperature for the Taq polymerase to start copying the strands. So coming back to the question, the 3 steps are: firstly, heating the DNA to 90 to 95 degrees Celsius to separate the strands. Step 2 is to cool to allow the primers to attach to the single-stranded DNA. And the 3rd step is to allow the Taq polymerase to copy the strands. Part C asks you to discuss the 3 factors that affect the migration of DNA fragments through the agarose gel during gel electrophoresis. Now, gel electrophoresis is a technique for separating fragments of DNA. The DNA fragments of varying sizes are placed on the agarose gel, and this is all placed in an electrophoresis bath where it is covered in a controlled pH solution. A power source is attached to the electrophoresis bath and it is switched on. DNA being negatively charged will move through the gel bath towards the positive terminal. The gel acts as a molecular sieve, and the smaller DNA fragments will move faster through the gel compared to the larger fragments. So based on that knowledge, here are some of the facts that you can use for your answer. Obviously, the size of the molecule is important. The larger the molecule, the slower it will migrate through the gel. The concentration of the agarose gel is also a factor, as the denser the concentration, the slower the movement of DNA fragments would be through it. The charge of the DNA fragments will also play a role, as they have to migrate towards the positive terminal. And finally, how long the voltage is applied for will also play a role, as the longer the voltage is applied, the further the DNA can migrate through the gel. Part D asks you to describe one ethical and one social issue that could arise from finding out if a couple has the same severe or deadly mutation, and here are a couple of sample answers. Question 9 talks about the Zika virus as a rapidly emerging viral disease, so pause this video to read through the question stem and recommence when you're ready to move on to the question. Okay, Part A asks for one social or economic factor that could lead to the transfer of pathogens to humans from an animal host. Essentially, more humans will need to be in contact with the animals to increase the chance of a transfer. Therefore, if you increase the population spread to areas of wild animal habitats, the transportation of wild animals to urban areas, any of these answers will do. Part B states that identifying antibodies to the Zika virus is challenging, as the test could not always identify the difference between the antibodies produced against the Zika virus compared to those against other viruses. The question then asks you to explain why making a correct identification of a viral pathogen is important in the control of a disease. This is a 3-mark question, so you do need to make multiple statements relevant to the question. In my answer, I mentioned that the correct identification of the pathogen would allow the scientists to isolate the antigen. That's statement 1. This allows the creation of a vaccine or drug to target the antigen, which is statement 2. And then this would allow the majority of the population to be vaccinated or treated, and that's statement 3. To be honest, my final sentence is probably a bit of a repeat, so technically it's probably not required. Part C asks you to explain why the antibody test could not identify the difference in the antibodies produced against the Zika virus compared to other viruses. The question highlights that in your response, you must refer to the structure of the antibody. So you need to mention that the viruses would bind to the antigen binding sites of the antibodies. So you can assume that the shape of the Zika virus antigen must be very similar to the antigen of other viruses, and as a result, they can both bind to the antigen binding site of the same antibody. Part D talks about the Aedes mosquitoes not being found on every continent, and they can fly great distances. They cannot fly great distances, sorry. The question asks for 3 different approaches that the government health officials could use to reduce the spread of the Zika virus. I've listed a few measures here. Obviously, you can have some other examples to the ones I've listed. But essentially, any measures you can think of that minimizes the spread of Aedes mosquitoes, or minimizing the spread of disease from mosquito to humans or human to human transmissions would be correct. Now, I couldn't access the articles for question 10 due to copyright reasons. Although you can access the full articles that the question was sourced from, I'm not sure what excerpts were used, and therefore I don't know what the questions are asking. There are some parts of question 10 that are knowledge recall, such as mitochondrial DNA versus nucleus DNA, but I didn't think I would be able to go through the entire question with you guys, so I'm going to move straight on to question 11. Now, question 11, which is the last question, is worth 11 marks in total. This is a long question stem, so please press pause and recommence when you're ready to move on to the questions. Okay, Part A asks you to analyze the table and state at which temperature was the measurements the most precise. And the answer here is 35 degrees Celsius, as the variations between the adults between the results was the smallest at just 1 second. Part B asks you whether this data is qualitative or quantitative. It is quantitative as we're measuring numbers, and it asks you to outline the difference between the 2 types of data. Quantitative data is measured numerically, whereas qualitative data is expressed descriptively. In Part C, student number 2 repeated the experiment and their results are displayed in Table 2. He indicated that he had errors in his measurements, and the question asks you to A, identify which of his measurements have errors, and then B, to give 2 examples of sources of errors for these measurements. Now I've put table 1 on the side for comparison, and as you can see, the two errors were made was measurement 2 for 15 degrees Celsius, as well as measurement 3 for 25 degrees Celsius. In both of these cases, the result is much lower than the other results, which means that it took a lot less time for the solution to become neutral. Things that could have been done to give these inaccurate results include adding too much lipase to the solution, incorrectly recording the time, or that there was just less sodium carbonate in the test tubes to begin with. Part D had student number 3 performing an experiment, and their results are listed in table 3. When he compared his results, he realized that he made an error. Name the type of error this student made and identify one source of error that may account for the results obtained by this student. As you can see, all of the numbers are consistently lower compared to the results of the other 2 students. This is called a systematic error, and one source of error could be that the student was using the measuring cylinder incorrectly and just adding the wrong amount of lipase to each test tube. Other answers could be equally acceptable, but this is just an example. And the last question, part E, asks you to identify a limitation of the experiment that was carried out by all 3 students and what they could do to address this limitation. Here you could mention the fact that there is no control design for this experiment, and the students could address this by adding a control arm where no lipase is added into the test tubes. Other answers could be that the temperature was only tested from 5 degrees to 35 degrees Celsius, and the students could increase the range of the temperature that they test. But this is just an example. So there you have it. That's my walkthrough of section B for 2019 VCE Biology Written Exam. I hope you found it useful. If you did, remember to give this video a thumbs up and subscribe to the channel, as I'll be posting more walkthroughs onto this channel, as well as other educational and studying videos, I think. If there were anything that I didn't cover in great detail that you want me to explain, or any other queries, please leave it in the comment section below and I'll do my best to get back to you. Thanks for watching, and until next time, remember to keep calm and study on.